Protein synthesis.
Talal, Vincent.
Ali.
Transcription is the first step in protein synthesis, and it is the process of making an mRNA code from DNA. The RNA differs from DNA in two ways. The sugar is ribose instead of deoxyribose, hence RNA instead of DNA. Also, there is no thymine in the RNA and there is uracil instead. There are three parts to transcription: initiation, elongation, and termination. During initiation, the transcription begins when the RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. Promoter regions are short sequences of DNA usually found right before the coding region. A subunit of the messenger DNA called the sigma factor takes part in recognizing the promoter region. Next. During elongation, the RNA polymerase moves along the DNA double helix and unwinds the helix as it goes. Then it begins to synthesize a complementary single-stranded mRNA using the DNA coding strand as a template. Soon after transcription begins, the sigma factor disconnects from the RNA polymerase, and next the RNA polymerase unwinds the DNA and the RNA polymerase will assemble bases that are complementary to the DNA strand being copied. This happens in the 5' to 3' direction. The sequence of nucleotides on the mRNA are complementary to the sequence of nucleotides on the coding strand, except that uracil is substituted for thymine. Afterwards, termination occurs when the RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the stop codon. A hairpin loop structure forms in the mRNA, causing the RNA polymerase and the mRNA to detach from the DNA. The mRNA produced is called an mRNase transcript. This marks the end of the transcription process in protein synthesis.
Once transcription is finished, the mRNA leaves the nucleus and attaches itself onto a ribosome. A group of 3 bases is called a codon, and for every pair of codons there is an anticodon contained on the tRNA, also known as the transfer RNA. It is called the transfer RNA because it also contains an amino acid for every corresponding codon. The first codon on the mRNA will be AUG, which is the start codon, and the anticodon for AUG is UAC. The tRNA molecule containing the anticodon UAC will attach itself onto the corresponding codon AUG. It will bring with it the corresponding amino acid for AUG called MET.
So at this point in the translation process, after the tRNA molecule containing UAC has attached to AUG and the translation process has, by a term, started, now the other tRNA molecule will come in, which would be complementary to the GCG base sequence. So the tRNA molecule for this would have an anticodon of CGC. The tRNA which contains this anticodon will also have a corresponding amino acid, ALA, or ALA, or however you pronounce it, and it will now go and attach to the codon. So once again, CGC, the anticodon, will attach to GCG of the mRNA, the codon. Once its attachment is completed, the two tRNAs will now be next to each other, and so a bond between the amino acids MET and ALA form, and this bond would be a peptide bond. After said process, the mRNA shifts over to the left of the ribosome, essentially leaving out the AUG. And at this point, the tRNA, which originally contained UAC, can now detach itself from the ribosome and the mRNA. But its MET amino acid is still attached to the ALA amino acid through the peptide bond. Now, because mRNA has shifted over to the left, the next codon, which is UCA in this example, has come in the center, next to GCG. So UCA, uracil, cytosine, and adenine would have a complementary anticodon of AGU. So AGU would, whatever tRNA has the anticodon AGU, would also have the amino acid SER. So this tRNA will now recognize that it needs to go to this codon, and it will go and attach itself to the codon, and the anticodon and the codon will pair up with each other because they're complementary to each other. And because they're now next to each other and they're attached, there will be a new peptide bond between ALA and SER. So now we have a string of MET, ALA, and SER. So at this point, we have 3 over here, MET, ALA, and SER. Now after this, once again, because now the 2 have been attached, once again the mRNA strand will shift over to the left. And now it'll introduce the UGA codon into the ribosome. And at this point, the UGA is essentially the stop codon. It tells the process to essentially do it, to stop. So UGA would have a complementary anticodon on the tRNA. It would be ACU. ACU has the amino acid which tells everything to stop, as I mentioned before. So this tRNA here, as I've drawn, would now go and attach to this. All this part over here would transfer over to the left. This tRNA molecule would detach like this one, but leaving behind MET and ALA attached. SER here would move to where ALA is, and this tRNA would come and attach here. But at this point, instead of UCA will now go over here, and UGA from here will now go to where UCA is. The tRNA will now come follow the UGA codon, and it'll attach itself here. The bond will be formed, a peptide formed between SER and the stop codon will be formed, and then the process will essentially be over, and all the tRNAs can detach, and now we have a string of amino acids called a polypeptide, and this can then be used for whatever the purpose be.